اوكي دكتور، اذا تحبون نبدي هسه.
إن شاء الله. إذن على بركة الله نبدأ. بسم الله الرحمن الرحيم، والصلاة والسلام على محمد وآله وصحبه الكرام المنتجبين أجمعين. تتشرف الجمعية العراقية للسموم الفطرية والملوثات وبالتعاون مع الجمعية العربية للحفاظ على الفطريات باستضافة الأستاذ الدكتور أحمد محمد عبد العظيم الغني عن التعريف. الأستاذ الدكتور أحمد محمد عبد العظيم أستاذ الفطريات في جامعة قناة السويس، مؤسس ورئيس مجلس إدارة الجمعية العربية للحفاظ على الفطريات، عضو منظمة الفطريات الأمريكية والأوروبية، عضو المكتب التنفيذي للرابطة الدولية للفطريات، مؤسس يوم الفطريات في جمهورية مصر العربية، نشر أكثر من 85 بحثاً وشارك في كتابة 50 كتاباً ما شاء الله، وألف 9 كتب، ورئيس تحرير مجلة الميكروبيال بايو سيستمز. إذا أتحدث عن إنجازات الأستاذ الدكتور أحمد الحديث يطول. أستاذ نرحب به، وتفضل أستاذ لتقديم محاضرتك، وأهلا وسهلا.
السلام عليكم ورحمة الله وبركاته. أهلا بحضراتكم، أخونا الفاضل أستاذ دكتور عادل وأخونا الفاضل دكتور سرهد، وبشكركم جدا على المجهود الرائع اللي طالما غمرتمونا بكرم أخلاقكم في الدعوات للحضور لهذا المنتدى العظيم اللي بيضم مجموعة من الباحثين في دولة العراق وبعض الدول الأخرى لإسراء العلم ونقل الخبرات والتبادل بيننا وبينكم. لكن خلوني في الأول أنا قبل ما أبدأ المحاضرة بتاعتي أتقدم بخالص العزاء طبعا لأهالينا في دهوك للقصف العدوان الغاشم اللي أطال الأبرياء من الجانب التركي خلال اليومين الماضيين، وبأسف يعني لما حدث، وندعو للشهداء والمصابين بالشفاء والشهداء بالرحمة والمغفرة. خلوني النهاردة أبدأ أتكلم مع حضراتكم في نقطة مهمة جدا قد تكون مسار العديد من البحوث خلال 10-15 سنة اللي فاتت، وسعيد الحظ إني تعاونت مع إخواننا في العراق وتعاونت مع باحثين من اليانج انرجيتك جينيريشن من الإخوة في العراق والأخوات في العراق. وطبعا بذكر من دهوك، بذكر الدكتورة تيروز والشغل بتاعها وحصولها العام الماضي على جائزة الأشيفمنت للجمعية العربية للحفاظ على الفطريات عن كم الإنجازات اللي ابتدت تعملها وهي لسه شابة صغيرة وطالبة في درجة الدكتوراه، ومجهودها الرائع في إنها تكون قائدة في موضوع الاندوفيتك فانجي وكيفية الحصول على فطريات منها لها القدرة على إنتاج مجموعة متميزة من جزيئات النانو بارتكلز، وبتقود بكده فكرة ومدرسة جديدة ليها إن شاء الله تعالى في العمر الطويل ليها ولحضراتكم في دولة العراق الشقيق. المحاضرة بتاعتي النهاردة يمكن أنا ألقيت المحاضرة في خلال زيارتي الأخيرة لجامعة بروجا. أنا كنت سعيد الحظ خلال المرحلة اللي فاتت في أن أنا كنت في زيارة لدولة إيطاليا، وتمكنت من زيارة العديد من الجامعات الإيطالية خلال الزيارة، جامعة تورينو العريقة، جامعة برما، جامعة باري، جامعة بروجا. فمن الجامعات العريقة اللي لينا تعاون معاهم ومهتمين جدا هم بوجود مجموعة من البايو اكتيف ميتابولايتس اللي بتطلع من الفنجي وتطبيقاتها، سواء كانت على مستوى الفارماسيوتكس أو في مجال الفود مايكروبيولوجي، وتطبيقاتها في إيجاد مجموعة من الفطريات لها القدرة على إيقاف الفطريات المنتجة للسموم الفطرية. المحاضرة تحت عنوان Endophyte Bioprospecting for Antimicrobial Metabolites، والموضوع زي ما قلت لحضراتكم بيحوز وبيستحوذ على انتباه واهتمام السادة الباحثين في ليست في جمهورية مصر العربية فقط لكن على مستوى العالم. وبدأ الاهتمام بالاندوفيتك فانجي في بداية عام 1990 من القرن الماضي، فاحنا بنتكلم على كائنات بتدرس لأكثر من ما يقرب من 30 سنة، بقالها فترة كبيرة جدا بتدرس. فمهم جدا لينا ان احنا نركز على هذه الكائنات. مش هقول انا مين لان اخويا الدكتور عادل استفاض في تعريفي، لكن خلونا نركز في الكام تعريف المهمين جدا اللي لازم اي باحث في الفنجي يعرفهم. What is meant by plant resident microbiota? The organism which get established and adapt themselves on their plant habitat and able to multiply on their own. التعريف ده تعريف عام جنرال بيحط لي هنا الكلام له علاقة بالميكروبيوتا. فيرد عليا الدكتور عادل يعني يقول لي ممكن ينطبق على الاندوفيتك بكتيريا. Yes professor you can say it for endophytic bacteria. Endophytic actinobacteria, yes endophytic actinobacteria. Endophytic fungi, yes endophytic fungi. So microbiota related to plants are broadly classified into two categories, epiphytes and endophytes. So epiphytes are the microbiota which localized on the surface aerial part of the plant, which contains leaves, shoot, flowers, fruits, whatever. It's called epiphytes, while endophytic fungi, it should be localized inside the tissue of the plant without causing any harmful effect, without showing any pathogenic symptoms. Because if it showing pathogenic symptoms, it is not endophytic organism, it's called pathogen. So there is a big difference between endophytic fungi, endophytic bacteria, and the pathogenic fungi and the pathogenic plant pathogenic, plant pathogenic fungi and plant pathogenic bacteria, because both plant pathogenic fungi and plant pathogenic bacteria should be showing what we can say it's pathogenic symptoms causing disease, but endophytic organism showing nothing. It's living without any symptoms or disease criteria on the plant. The terminology, as I mentioned before, endophytes is microbiota inhabiting the interior part of the plant, especially leaves, branches, root, and stem, without any harmful effect to the host. So Adil asked me or any one of you asked me, Ahmed, then when we go to select our sample to isolate endophytic fungi, we should select the healthy part of the plant? Yes, you should select the healthy part of the plant. You must select the healthy part of the plant, not any diseased part, because it is not good for your research on endophytic microorganisms. While epiphytic or epiphytes are microorganisms inhabiting the area of the aerial part of the plants, and this is what we already Azevedo in 1998 identified them. But we can say microorganism growing on the surface of photosynthetic organisms. So some guys said, what about seaweeds, الأعشاب البحرية? If the organism inside it, what we can say that it is endophytic microbes? Yes, it's endophytic. And we can call it algicolous fungi. What about the surface? Yes, you can say it's a phylloplane or phyllosphere microbiota. So the microbiota surrounding the aerial surface and the aerial surface of the leaf or air surrounding the leaf, it's called phylloplane and phyllosphere biota, either fungi or bacteria. So Henrik Anton de Bary who is already first mycologist. He's a botanist and mycologist, and he is the first person all over the world coined to terminology related to epiphytes and endophytes all over the world. No one knows the two terminology before D. Barry. So what you can say about the biological characteristic features of the endophytes, you should know how they can penetrate the plant. We have, based on their behave or their entry, we have vertically transmitted endophytes or horizontally transmitted endophytes. So vertically transmitted endophytes, they came directly from parents, came from seeds, came from the offspring, and still sterile, and we can doing that easily if you have some grains of wheat and you can doing self-sterilization of the wheat and or corn seeds or corn grains or fiscia faba seeds, and you can just self-sterilizing them and put it on culturing media. You can recover many endophytic fungi which already genetically inherited with the offspring. They are transferred from offspring to the another due to their vertically transmission. While horizontally transmitted, which already came from soil, came from insect or aerial dispersal. Oh my God, how it comes some endophytic fungi or endophytic bacteria are horizontally transmitted. They are transmitted through the absorption of what we said soil solution, which already covered with them, which already contains a nutrient, salt nutrient, and the spore of fungi, and absorbed by root hairs, and from root hairs it directly transferred by xylem and located in the stem and or stem root zone, transition zone, or on the leaves or to the fruits. So it's horizontally transmitted. So based on the entry pathway of the endophytic, we have two big categories: vertically transmitted endophytes or horizontally transmitted endophyte. And sometimes also during the horizontal transmission, it transferred from air. Oh my God, how it comes? It's transferred through stoma, sagr. So through stoma, some spores can enter the plant and localize there and transferred horizontally, making a colonization of endophytic fungi or bacterium. So this is one of Truja contribution in our new book. How can she already express the transfer of endophytic, either vertical or horizontal transmission? This is give you an illusion or just an illustration to how can this microorganism go inside the plant. How they can penetrate the plant. Is it come from directly from the grains or the seeds, or they come from indirectly from soil? I mean vertically or horizontally. On the other hand, we can classify endophytic fungi or microbiota into two big categories. The first one, it's called systemic endophytic or endophytes, and the other class is called non-systemic or transient endophytes. So systemic endophytes, which live within plant tissue for the entirety of its life cycle and participate in symbiotic relationship without causing any disease, without showing any pathogenic symptoms. So it's called systemic endophytes. While non-systemic or transient endophytes, it's a vary in number and the diversity within their plant host under changing of environmental condition. What is meant by changing of environmental condition? Yes, you can find, as example, Artemisia herba alba plant. You can get some of its endophytes on a mountain at elevation of 1,500 meter above sea level. And after you go for 2,000 meter above sea level, you can't find this endophyte inside it. So based on its environmental condition, it's called non-systemic. On the other hand, some taxa of endophytic fungi, which we are considered it safe under some environmental condition, looks like climate change, increase of ultraviolet radiation, it's converted directly from endophytic into pathogenic. So that is why we are keeping all the time said in all international meetings, please take care of fungi because it's converted from endophytic fungi, from peaceful organism into dangerous one, which already causing a lot of diseases of plants due to climate change. On the other hand, there is a biblic paper related to classification of endophytic fungi based on their taxonomic level, on the taxonomic rank. It's called Rodriguez et al., which already those authors classified the endophytic fungi into two big categories. The first class is called C. endophyte or Claviciptious endophytes. Claviciptious endophyte or class number 1, they are small group of fungi phylogenetically related to Claviciptious species, which already located in phylum Ascomycota, and they are nutritionally difficult to cultivate. It's a very, very hard group to cultivate them in our laboratory. Sometimes we can isolate them by a special technique, but these organisms is a very, very, very potential organism. You can use them in different applications related to agriculture. Looks like increase the production of wheat crop, corn crop, anything related to production of grass crops. So you can use Claviciptious endophytes. On the other group, non-Claviciptious endophytes, which contains the other groups of Ascomycota and some Basidiomycota with few number of Zygomycota. So it's able to found easily in all the plants. So Adel or Serhet can isolate some fungi from a plant in Iraq, and they can found the same species in the same species of fungi in the same species of the plant in Egypt, because they are very, very common and very easy to isolate them, and they are considered different classes based on their colonization. So we have class number 1, Claviciptious endophytes, and the class number 2, class number 3, and the class number 4, they are belonging to non-Claviciptious endophyte. And as I mentioned in this slide, Claviciptious is a big group in Ascomycota. So it's a kind of taxonomic classification of endophytes. And this is a table who already Rodriguez et al. Mentioned them in 2009, and you can go through the different criteria which they classified endophyte into two big classes, Claviciptious and non-Claviciptious. So Claviciptious is just only one class, while non-Claviciptious is classified into 3 classes, class 2, class 3, and class 4. What about the host range? Host range related to Claviciptious fungi are very narrow. They are very, very narrow. It's just located in some grasses, looks like Tifa and BOA, and it's a very narrow hosting the plant. They are colonized in shoot and rhizome. It's able to doing extensive colonization, but for these species, but very low in comparison with the biodiversity of class number 3 of non-claviceptious. Their transmission is either vertical and horizontal, and the fitness benefits of them, what we call the non-habitat adapted, which already doing what we said, a very potential benefits to the plant, such as drought tolerance and the growth enhancement. They are very potential taxa. You can bring them from India and add them to your plant in Iraq, in Egypt, in Tunisia, in Algeria, in Pakistan, and they are working in a great way. On the other hand, the different classes of non-claviceptious, they are colonized, all of them are broad spectrum. They are found on the majority of the plant. And tissue colonized is in the class number 2, shoot, root, and rhizome. In class number 3, shoot. In class number 4 is root. Extensive, class number 3 is very limited. Class number 4 is extensive. So this is a comparison between what we said, how we can use this endophytic fungi, class number 2. You can use it in the habitat adopted benefits result from habitat specific selective pressures, such as pH, temperature, and salinity. So if you are going to design your paper and your work to isolate endophytic fungi to enhance salinity, you should working on class number 2. Okay, but if you are going to work on and pH sure, and if you're going to doing some other studies, you can select between class number 1 and the class number 2 and 4. Here, what we said, a diagrammatic representation of endophytic fungal association. So some organisms came from soil as a saprophytes and gona inside the plant, and they still as saprophyte. Other gona as saprophytes, and sometimes they are some species, I mean, gona as in endophytic fungi and inherited and transferred as endophytic fungi. Some kinds are coming as pathogenic parasites, and after they go inside the plant, the internal structure of the plant and the chemical constituent of the plant converted them directly into endophytic. They made them latent. And you can find, based on our researches and studies, some endophytes are able to convert it into pathogenic fungi due to what we already called, what we already discussed before, climate change. So you should know that is a fungi or endophytic fungi born to the tissue by different methods. Looks like it can able to forming intercellular mycelium. The mycelium of the fungi is running between cell, or intracellular mycelium, which already inside the cell, or inter and intracellular mycelium. This is the main invading way of endophytic fungi related to plants. There is different mechanisms related to interaction of endophytes with their host. So endophytic fungi doing a lot of potentiality for the plant, and you should summarize that in the following figure. You can find the plant is affected in a positive way by the presence of endophytic fungi. Oh my God! So the plant is not free of fungi or not free of bacteria or actinobacteria all the time, as we imagine. It is not sterile. It contains a lot of microorganisms inside it to help it during its growing up to frotting. So endophytic fungi can increase abiotic stress tolerance, as we see in this part, increase efficiency of antioxidative system of the plant, manipulate proline and flavonoid content, modulate phytohormone levels. So this is a part related to fungi. Increase nutrient uptake as increase root development. You can find the plant was invaded with endophytes. It's characterized by what we said, various and strong secondary root and root hairs, upregulate metabolism and the transporter gene, produce exolytic enzymes and metal chelators. So it's all the time it's produce a lot of materials support the nutrient uptake of the plant. On the other hand, modulate photosynthesis, which already increase chlorophyll content, reduce leaf fiber conductance, affect internal carbon dioxide concentration and the carbon dioxide fixation. As we told before, that is fungi and the presence of mycelium, even in the plant or inside the first 10 centimeter of the soil, it captures carbon dioxide in the form of polysaccharide from the soil, and about 75% of the carbon dioxide all over the world is captured in the first 10 centimeter under the ground by a mycelium network formed by fungi. So it's already affects the internal carbon dioxide concentration and the carbon fixation. What about increased biotic stress tolerance, increased lamina or leaf area, affect the physiology of herbivorous insect? Some endophytic fungi produce VOC compound, volatile organic compound, which is able to kill insects. Some of these compounds completely are volatile and able to kill insects. Also, effect of physiology of herbivorous insect, promote jasmonic and salicylic acid pathways, which already doing a great work for the plant against biotic stress. How we can say there is an interaction of endophytic fungi with their host. So the relation between fungal plant interaction takes place during the seed or seedling or adult plant, or even during the fruiting of the seed. How this endophyte is transferred from the seed, which already what we said, it's a vertical transmission, and he already in a temporal residence transferred after that into form of seedling, it germinate, and the rhizosphere potential of endophyte support the rhizosphere, and it make a lot of biotic help for the plant. After that, endophyte is transferred to the tissue through the xylem vessels and give it the plant potentiality against pathogenic fungi and blah 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 blah. So there is a big relationship between endophytes and what we said, the plant life cycle. All plants are infected with microbes, and we said symptomless is epiphytes, endophytes, and it's a kind of balanced status of symbiosis. And this is happen in the majority of the plant, or it's caused a disease caused by pathogen, and this is considered unbalanced status of symbiosis. What is the gain of endophyte from the plant, and what is the plant cost? The endophyte gains shelter and the nutrient, and the plant cost nutrient and resources, and plant gains growth promotion, increased tolerance to harsh environment, induced resistance to pest and disease. And we already have one of our young student in my laboratory. It's called Fatma Bunouh. Fatma will defend her master degree soon on using endophytic fungi to enhance tomato production under abiotic stress of salinity and drought. So her work is all the time designed to select plethora of different categories of endophytes, and how can she re-enter them into tomato either by seed or seedling or by spray for the adult plant to select the most suitable way to enhance the production of shoot system and the fruit and chlorophyll amount increase in tomato hosted by endophyte in comparison with normal tomato. So why we selected endophyte in our research? What we should say of the 300,000 known species of plant, there is at least one plant associated endophyte. So if we suggested the plant species Oliver's globe is about just 300,000 species, and you can say inside each plant is just one species of endophyte, so you will have already 300,000 isolate of fungi. Conservation of endemic and medicinal plant, as I told in many presentation before, if you go to collect a very potent medicinal plant to extract 0.0 gram of volatile oil or a drug for your research, and you can discover later on that this plant contains a fungus which produces the same drug. So why I'm going to over collect this plant and destroy the environment and the protected area? And this is not, it's considered prohibited in a lot of countries due to conventions of biological diversity, which already prevent all the over collection and deterioration of the plants as example. So also the WHO et al, they are a team of researchers in China. In 2010, they published a nice work considered endophytic fungi for producing bioactive compound originally from their host plant. Oh my God. So the endophytic fungi, a fungus, which you already recovered from the plant, you can find it produce anti-cancer material if your plant is producing anti-cancer material. What about if you are working on a plant producing antimicrobial? You will find your fungus producing antimicrobial materials. So this is happened due to co-evolution and residence of the fungus inside the plant. There's a kind of genetic transfer between the plants and the endophytic fungus, and this is gives the gene of the endophytes the ability to produce the same material of their host. On the other hand, there is a lot of control to over collect plants and animal, even fungi, all over the world. So why you can go to collect or over collect a medicinal plant while you can isolate a fungus inside this plant to produce the same material as the plant. Also, there is a list number of some natural product. Truges is doing this great work, which already showing us protozoa is producing about 100 up to 200 bioactive molecules. If you go about the scale, you can find increase of the needs of production of natural product, and you can find fungi are in the right now in the top for producing these bioactive molecules as in this part. plant kingdom, which contains part of bacteria, algae, and lichens. Higher plant, it's produced many bioactive molecules in comparison with fungi. Fungi is producing more than 2,500 thousand materials or drug all over the world. Some of them is related to endophytic fungi. This part is related to how you can isolate endophytic fungi. For isolation of endophytic fungi, you can start first by histological observation. You can doing what we said, surface sterilization. You can run and can collect your a part of this plant and take it the plant, the healthy part of the plant, into your laboratory, and you can cut healthy leaves into small pieces, and also the stem, and doing the surface sterilization technique. This technique is based on running in washing and running water, surface sterilization by chlorix or sodium hypochlorite, and after that by ethanol, and incubate your pieces on different isolation media. And after that, when you got your cultures, you can transfer it into slant, and this is based on Abdelazim and the Salim technique in 2000. On the other hand, there is a kind of serial dilution technique. Serial dilution technique is based on a technique suggested by McNorrie and Klober in 1995. They are using a surface sterilization technique, and after that, they take this part and blending it. It can use a blender in a sterile condition, in a sterile solution, and take it the volume of it to use a bore plate technique. This is, from my point of view, it's advanced technique for a people, because it's if any mistake will happen, you will lose all your petri dishes. You should start with a surface sterilization technique as an easy technique, and after that, if you are good and they are expert, you can transfer to serial dilution technique. So the question right now, how you can select your target plant? Abdelazim et al. Is suggested 3 points related to your target plant. For isolation of endophytes, you should select unique environment. It is not recommended for you to go to some grasses and some trees in a common garden to select the tree for isolation of your endophyte. You should select unique environment, a top of mountain, a desert without any disturbance, some salt marshes area, some unique environment. So when you select your target plant for isolation of endophytes, you should select it in unique environment. Number 2 is diversity. Diversity is meaning a hot spot for diversity. It's not recommended for you to go plant population. Not work on a plant population because it's just one species of plant, but you should select a plant community. Different species, you can select from species number A, species number B, C, D, E. You can select different diverse species of plant and transfer them to your lab to isolate your endophytic fungi. Ethnobotanical history and endemicity. This is mean if you are working for Baghdad or Dahouk, so you can go to the herbicists there and ask them about the history of this plant. Is this plant is used in folk medicine? Yes. How many years it used? Oh my God, it's 1,000 years used in folk medicine. Okay, you can select this plant to isolate your endophytic species. So this considers the most important 3 criteria for selection of your target plant for recovering or isolation of endophytic fungi. For myself and my team, we already started the research in St. Catherine area, and St. Catherine area is located at South Sinai, Egypt, in Sinai Peninsula, and it contains one of the largest protected area in Egypt, about 4,500 square kilometers. And we started our researches maybe in 2003. So doing a lot of researches concerning endophytic fungi. The study area is, as I mentioned, it's located in South Sinai, and it's a mountainous area. And all of you know St. Catherine well, and it contains a lot of wadis. These wadis contain some very potential medicinal plant. Besides, they are endemic plant. So they are endemic, and they are already very characterized by their ethnobotanical uses by local Bedouin in this area. And also you can find the different elevation wadis or different elevations start from 1,400 meters above sea level up to 2,362 meter above sea level. So different elevation, different vegetation, different ecotypes of the same species, you can collect your plants. We are, as example of one of our study, we selected 9 or 8 species from the endemic species. They are used for a long time in folk medicine, and we directly started to transfer to our laboratory for isolation. Here is what already said Abdelazim and Salim in 2012 surface sterilization technique, which already based on take a plant sample, washing it in running water for 10 minutes. After that, you can cut it into small pieces, not more than 1 centimeter, and you can at first part put it in ethanol, or sometimes I prefer to start with sodium hypochlorite or chlorix. You can start with ethanol for 1 minute or 30 seconds based on the thickness of the plant. And after ethanol, you should go directly for distilled sterile water to remove the remains of ethanol. And after that, transfer your segment directly to the chlorix up to 5 minutes based on the thickness of the plant. If you find that it's woody, you can put it for 5 minutes. If it's herbaceous, it's just 30 minutes or 1 minute. And after that, after chlorix, you should wash it by distilled sterile water. And after that, you can transfer it to the ethanol. And after ethanol to distilled water, dry it on sterile filter paper, and incubate it on a petri dishes. As you saw, the pieces should be 4 parts away from each other to give the fungus the opportunity to grow. And you can use different isolation medium. Looks like looks like Chabex yeast extract agar, botulinum agar, malt extract agar, whatever. But your media should be supported or supplemented with rosbengal as a bacteriostatic solution, and chloramphenicol as a bactericidal antibiotic to suppress bacterial growth, because you are running after fungi. You are not running after bacteria. And this is different initials of media, Chabex yeast extract agar, BDA, MEA, potato carrot agar, and oatmeal agar. Some of our fungi growing in petri dishes. Here you can notice that the fungus is come directly from the plant segment. It is not growing outside the plant segment. If it's growing here, it is not considered endophyte, it's considered a contamination. And you can doing a kind of experimentation to be sure from your sterilization technique, as you can keep the last step here of water here, and take from this 1 drop or 2 drops and disperse it on a petri dishes. So it's suppose this is without any spore of fungi, because it came surface sterilized to this number 3 beaker number 3. It should give nothing on petri dishes at all after incubation for a week. After that, you run what we said identification, and the identification in our laboratory, we used phenotypic identification based on morphological criteria under microscope, dimension of the spore, conidiophore, branching, shape of conidiophore, color of conidia, whatever, by using different relevant description keys. Looks like compendium of soil fungi, atlas of soil ascomycetes, Dematteia cyphomycetes, whatever. If you are working for fungi, your lab should contains a lot of updated keys. It is not allowed for you in 2022 are using a Gilman in 1940 as a key for identification of fungi. Oh my God, it's not believable at all. You are in 2022 and all of these taxa completely, some of them synonymized or changed into new species, so you should update your information. And the majority of fungi are belonging to different phyla, but as I mentioned before, in endophytic fungi, we should obtain either Basidiomycota or Ascomycota. If you found Mucoromycota or Zygomycota, it's a very rare, very rare phylum recovered in endophytic fungi. So take care. If you find in your work some Rhizobus, you should check your surface sterilization technique. It may be contamination. Okay. You can doing a molecular identification by different 18S ribosomal DNA techniques, by DNA extraction. If you can using some DNA extraction kit, but I think we are preferred to use a modified technique suggested by Blanchett et al. In USA. We are using it for extraction of DNA. It's much better for if you are going to extract your DNA. It's much better from all kits in the markets. So after that, you can do PCR and send your sequence into a sequencer in Korea or something like that to get your peaks, and AB1 file doing your revision of the genes, what is missed, and you can doing after that FASTA and applying your sequence into a gene bank. Your identification of a species, it's based upon your money. I mean, if you isolate 70 species of fungi and you are not good in identification of fungi, so you are going to send them all the 70 for identification outside the country or in a special lab, and you will pay a lot of money in front of this. And for my laboratory, we are just working on confirmation of the potential taxa. If we isolated 70 species, we are doing examination by microscope. I am able to identify them up to species level, and we can select some of them and doing a more investigation on them. And after that, we can just molecularly confirm the species. It is not identification for us, it's just confirmation. If you said it's Aspergillus niger or brasilensis or Aspergillus japonicus, we just send it for confirmation. And after that, you're doing a lot of statistical analysis, looks like multivariate analysis, twin span or Canuco, whatever. and the descriptive statistics through ANOVA by using SPSS. Okay. What is the mechanism of attacking enemies by endophytes? The endophytes alter the disease development in plant directly or indirectly through, number one, direct inhibition of plant pathogen by hyperparasitism. You can find inside your plant a fungus able to parasite the other plant pathogen which try to enter to your plant. You can find that easily. You can find some species of trichoderma, endophytic trichoderma, able to control the fusarium, some kind of chetomium able to grow on another kind of fusarium to parasitize it, some kind of acrimonium species or sarcoclidium able to stop it any pathogen. Also competition. The endophytic fungi will compete with the plant pathogen on the resources, antibiosis. The majority of endophytic fungi able to produce bioactive molecules, some of them antimicrobial. Also, endophytic fungi produce some lytic enzyme, looks like chitinase, and the chitinase able to degrade the wall of pathogenic fungi. So you have different ways. How can endophytic fungi attack enemies by mechanism? Or indirect effect by induction of plant resistance. It's able to enhance the plant resistance against the pathogen. Endophytes, as I mentioned, is doing a direct mechanism or indirect mechanisms. And here is the diagram is illustrated all the direct and indirect mechanisms referred to endophytic fungi or bacteria, which used to attack the plant pathogen inside the plant. As example, hyperparasitism, you can find a lot of endophytic fungi able to attack plant pathogen directly, as mentioned by Tripathi et al. In 2008. And they already recorded that by microscope, and they publish a lot of that. And you can find the mycoparasitism of Rhizocotonia solani by endophytic chetomium spirale as example. And you can find here the Rhizocotonia fungus is surrounded by chetomium, and after that chetomium is released its enzyme to degrade the Rhizocotonia directly. So no way it's a potential effort of the endophytic fungi to support his plant. Here is from the same study of Guyot et al. 2005 to showing the hyperparasitism between fungi of chetomium and how it can already release its decaying enzyme, and this is shown by TIM study. Competition, as I mentioned, endophytic fungi able to compete of other pathogenic fungi by different ways. Looks like utilization of nutrient, Cedrophore mediated competition for ferric ion. How can they capture ion from the environment and making it unavailable for the pathogen? It's a different ways. So I think if you like to read some of my recent publication, you can find it on Journal of Fungi, maybe this year, concerning endophytic fungi and weed plant, القمح. You can find it on free of, it's a free access, so you can find it on Journal of Fungi website. Fungal cedrophores production in vitro is affected by some abiotic factors. There is a lot of publication. You can find it all the time. Antibiosis, as I mentioned before, terpenoids, the compound, alkaloids, aromatic compounds, all the volatile compounds, VOC compounds are produced by endophytic fungi, showing what we said, antibiosis. It's able to kill plant pathogen. And here's some published great work concerning endophytes as a source of antibiotics. Antimicrobial metabolism from endophytic fungus Aspergillus versicolor as example. So if you like to run a research looks like that, you should select your plant, select your fungus, and run your research easily. Lytic enzyme production, a lot of endophytic fungi able to produce some potential extracellular enzymes, looks like cellulase, looks like protease, looks like amylase, looks like lactase, looks like lipase, chitinase as example. And you should take your attention that is when you recover different species of endophytic fungi from a plant or from different plants, the potentiality of each species is different from the other for production certain of enzymes. So you're not expected to isolate all Chaetomium species as endophytes, and you can find them produce cellulase as example. It's dependent on the species, on the environment, on the plant source. So here are some petri dishes showing the potentiality of some endophytic fungi for production of cellulase, lactase, tyrosinase enzyme, manganese peroxidase, which you can use it in wood degradation enzyme, whatever you like to apply your endophytic fungi later. It's able to also indirectly to induce plant resistance by systemic acquired resistance and induced systemic resistance by systemic acquired resistance already induced by pathogen infection, and it is mediated by salicylic acid and associated with the accumulation of pathogenesis related proteins. Some fungi is doing this kind of rule by increasing salicylic and jasmonic acid, which already start to enhance the plant resistance against the pathogen. So if the endophytes inside the plant will be ready, waiting for the pathogen to resist it. Here's a published work, 2006, Fusarium oxysporum endophytes induced systemic resistance against Rhodopholis similis on banana as a disease. And you can go through this paper. It's a nice paper. You can read that. Tam et al. Also list of endophytic species isolated from different hosts. And he used different taxa of Fusarium, and their host plant is banana, tomato, whatever. And he came, the sources came from Indonesia, Kenya, Uganda, Honduras, and use it for induction of systemic resistance. How can endophytic fungi work with another taxa? With another taxa, you can find or you can exploit or bioprospect endophytic fungi from lichens. We have a girl is called Safa. She is working on endophytic fungi recovered from lichens, as example in Nile Delta. So you can find endophytic fungi on the other microorganism. On the other hand, you can recover your endophytic fungi from seaweeds, from mosses, from pteridophytes, from gymnosperm. You can do that all the time. Here's one of my publication, assessment of secondary metabolites from marine-derived fungi as antioxidant. We recovered these fungi from seaweeds of Mediterranean Sea in Alexandria. Diversity of endophytic fungal associated with bryophytes. Soon, Me and my colleague Francesca Di Gola in University of Parma, we are already editing 2 volumes of a book related to endophytic and endolycanic, endobryophytic, and endotridophytic fungi, their taxonomy, ecological perspective, and application. So this will come soon, and Springer. 2 volumes will be nice for all researchers all over the world. Endophytes and the molecular studies, you can doing a lot of molecular studies related to endophytes right now in the genomic era. Looks like metagenomic studies, molecular markers, genetic expression, and molecular cloning. All these studies right now is very, very common. And you can go to any page in ResearchGate. You can find a lot of researchers doing many research related to molecular of endophytic fungi. How you can apply your endophytic fungi? You should put your target before you design your experiment. Are you going to work on phytosimulation? Are you going to work in enzyme production, pigment production, biocontrol agent, nutrient cycling, production of volatile organic compound as what we already said, VOC compound, source of bioactive and novel compound? Your experiment is your target. And based on your experiment or your idea, you can select your plant, you can select your taxon later on, because it's based on the literature, you can select your taxon also. And it's easy to select your taxon after you recover them from endophytes, from the plant. What we already have been done, myself and my great co-workers, we already published a lot of things. Screening of anti-cancer metabolites produced by endophytic fungi in St. Catherine protected area, Abdelazim and Salim. Evaluation of curative probability of bioactive metabolites, Abdelazim et al. Characterization of L-asparaginase produced by some Egyptian endophytes, Balbul and Abdelazim. Already different anti-cancer metabolites came from endobiotic fungus, Trichosessium roseum, and its biochemical effect as a strong candidate for MCF7 breast cancer cell line. You can find a lot of these researches. I already published 45 paper, book, chapter, project, and abstract within the last 10 years related to endophytic fungi. Some related to anti-rheumatoid, some related, as I mentioned, to L-asparaginase enzyme as anti-leukemic enzyme. Antioxidant. Right now, we have a girl working on in the end of her master degree. Heba Bunhas is working for antioxidant. Source of new pharmaceutical biomolecules, Muhammad Abdelazim and his colleagues. Also, Fatma Bunouh has write a good chapter on fungal antimicrobial molecules. And we already published, I already edited a big book related to recent development on genus Chetomium. Inside that, you will find many chapters related to endophytic Chetomium and how you can use this kind of chemical structure, looks like alkaloid ketamine, as antimicrobials. 2 volume is already produced related to industrially important fungi for sustainable development, biodiversity and the bioprospecting of biomolecule. Volume number 2 is already, both of them I have. If anyone interested to have them, he can or she can send for me a message on WhatsApp or email. I can send the book for you, and you can find a lot of interesting chapter. Here is our question right now. How can we set endophyte as an explored treasure to combat multi-drug resistance organism? we know that in 2020, a study carried by Sareja et al. Based on systematic online search showed that the evaluation of bioactive material produced from endophytic fungi present in the medicinal plant against multi-drug resistant microorganisms were considered for the review, and the period of study was limited to literature reported from 2010 up to 2021. The first literature search resulted 7,400 articles. And after that, you can find their study explored that the hidden potential of endophytic fungi as antimicrobial agent and found several candidates that can be utilized in the treatment of multi-drug resistant infection in human. Oh my God. So this research in 2022 said for us that your hidden treasure is endophytic microorganism. So why we are as a scientific community from Iraq, from Saudi Arabia, from Pakistan, from Algeria, from Tunisia, we are not looking after endophytic fungi. Why? I don't know. As I mentioned before, I can help anyone ask me help. But if you not ask me, I will not go to be a volunteer. So you can ask me help in that. We can apply together for international project, for cooperation, scientific cooperation, whatever you like, but just ask me and I will do that with a pleasure. And here is why, as what we said, that how can multi-drug resistant bacteria accept its resistance? High number of bacteria, with a few number of them are resistant to antibiotic. And antibiotic kill bacteria all the time because we are in a misuse of antibiotic in the majority of what we said in Arabic world and some countries in Europe also. So antibiotic kill bacteria causing the illness, as well as good bacteria protecting the body from the infection. When we are using misuse of antibiotics, the resistant bacteria now have preferred condition to grow and take over. Why? Because we are using antibiotics which already killed the beneficial bacteria, and after that, the space is right now is free for the pathogenic bacteria, which can take the opportunity to grow and to transfer their drug resistance to other bacteria, causing more problems. And I think all of us knowing this situation right now, if you're doing a study on, and I wondered, I'm a reviewer in many international journals, and I got a paper related to extraction of ethanol and from the higher plant, blah, 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 blah. Why you go to the higher plant? Why you collect huge amount of the plant? Why you destroy the environment to extract very few amount of the drug? You can go to endophytic fungi. You can do fermentation in your laboratory for 5 liters and extract whatever you want, and you can examine it. So if you're doing that, you will discover a lot of bacteria is able to resist your drug right now. So it's a big problem, and I think it will be soon as a future crisis for over the world after COVID. And here is a question for all of you. Do plant endophytic and the free-living Chaetomium species differ? If you isolate Chaetomium species or Chaetomium globosum from the soil as soil isolate, and you have Chaetomium globosum as endophytic fungi isolate, are there differ or not? Please think in that, and we can discuss by the end of the presentation. Different what we said, that's alkaloids from Chaetomium and more than 200 secondary metabolites belonging to genus Chaetomium And genus Chaetomium is a potential genus, is a potent genus in Ascomycetes in Egypt, and Ascomycetes in Iraq, and Ascomycetes in Saudi Arabia. Very, very important genus. Why? Because it's a cellulose degrading fungus. It's an endophytic fungus. It's a very, very potential for producing a lot of extracellular enzymes. So alkaloid compounds of genus Chaetomium looks like ketoglobosine, diketobibrazines, isoquinolones, tetramic acids, whatever. If you want to my book, genus Chaetomium, recent development, you can find chapter number 6 or chapter number 7 on Chaetomium alkaloid. This taxon, this organism, is a very important target right now for my research and my project, since projects already got funded on them since 2011. And I ask you to also concentrate on this genus. This genus is very, very important. You can find a lot of my contribution, and you will find in my book in the first chapter an updated key for all the taxa. I already prepared the key for the 300 species of Chaetomium worldwide. So you can use the first chapter in my book if you are looking for identifying Chaetomium on the phenotypic identification. So this genus is overlooked. Even in 1940, 1940, they found it produce antibiotic. This antibiotic referred to its alkaloids. So the alkaloids of Chaetomium can be used as antimicrobial substance. What we said, it's there is how you can discover your genome or your hypothesis concerning your gene producing this compound. So this is the best way, as example of some aspergilli. You can imagine how we can doing that, how you can analyze that even in a different basis. If you are start to doing that by phenotyping, typing, if you identify your fungus and you doing your research, and after you doing your research, you are select 1 or 2 species of these fungi against the Klebsiella, against MRSA, whatever, and you are going to doing a more analysis to select your product and how can its cycle or metabolic pathway is emerged, and you can run after what we said, synthetic genes clusters. Synthetic genes cluster is the target of many researchers worldwide, especially with pathogenic fungi. Looks like Nancy Keller in USA. She's running on pathogenic on the cluster genes, synthetic cluster genes against these pathogenic human fungi. Looks like Aspergillus tertius and Aspergillus fumigatus. Here is just a chart showing you how you can start with your idea, how you can doing isolation, and after that you can doing HPLC, doing bioassay guide, and after that you can run after your compound. Compound came from organism number 1, compound came from organism number 2, the same compound came from organism number 3, and doing what we saw, transcript analysis and PCR amplification and the whole genome sequencing. Right now they able to doing for you the whole genome sequencing by $3,000 or $4,000 US dollar. So you can doing that easy. So as I mentioned before, we are doing some phenotypic methods for examination your extract by phenotypic method, by dilution, diffusion, gradient test, chromogenic media, automated devices, and this take about 2 days up to 20 hours. And molecular-based method for PCR, qPCR, cycle sequencing, next-generation sequencing. It depends upon your money, depends upon your target. And after that, multi-TOF mass spectral analysis to examine your materials, your suggested antibiotic. So it's a scheme for all of you to doing to design your experiment for the antimicrobial. Our lab protocols is mainly on the localized in 5 objectives: isolation and taxonomic placement of endophytic fungi from unique plants in different ecological habitats in Egypt, antimicrobial screening of recovered endophytes, sequencing and bioinformatic identification of biosynthetic gene clusters of potent endophytic fungi with highest antimicrobial activity, fractionation, isolation, purification of antimicrobial compound from selected extract, structure elucidation of pure compound. This is not happen in this is objective not happen in, all in my laboratory, but we are doing that with other colleagues, colleagues in Egypt, colleagues in Italy, colleagues in USA, colleagues in Germany, colleagues in UK, in Scotland, whatever. But this is our main objectives to get our antimicrobial product. So you just here take an account about our protocol in our laboratory, just the fungal extract or fermentation, doing for some analyses for the metabolites, the whole metabolites, and the bioactivity screen. So after that, database and NMR for your extract to get biosynthetic gene clusters. So application of different biotechnological tools will help in strengthening and understanding of plant endophytic interaction, reducing the new bioactive compounds. And we should all the time and together to concentrate on our research on endophytic fungi. You can find the fungus able to inhibit the Aspergillus flavus produce mycotoxin. So you can use it in a dowel culture at first, and after that you can design a product for your feed for animal, so you can protect your animal by a simple endophytic fungi. And the last conclusion for my presentation, the microbe-free plant fact or artifact, sure, no microbe-free plant, sure, all plants contain inside of them endophytic fungi. So even if you're doing tissue culture, you will find inside the plant, inside your callus, is endophytic fungi. So it's a vertically inherited. Here is all my contact information. If you like to contact me or by email, by WhatsApp, by whatever you like. And thank you all, and thanks for Professor Adel and